Protozoa. In the domain Protozoa, we are going to see the phylum Plasmodiophoromycota. In the taxonomy and classification of fungi, I told that we're going to follow Kirketal classification, that is the 2008 classification based on the 10th dictionary of fungi. That is the most widely used classification at present. So now we'll see some common features of Protozoa and phylum Plasmodiophoromycota. I'm N.H. Shankar Reddy, PhD Plant Pathology. So this is the Kirketal classification, that is the 10th classification of fungi. So this domain Eukarya is actually divided into three: Protozoa, Chromista, and fungi. So we'll start with Protozoa. So what are the classifications and what are the detailed things will be there in Protozoa? Now we'll see. This protozoa was actually introduced by Goldfuss, George Goldfuss in 1818, and the taxonomy Protozoa erected at the time with a class with the Animalia, and it was erected with the Animalia. Later it was divided separately. Then this Protozoa actually derived from two Greek words, protos and zoon, which means first animal. This is believed to be the first animal that is originated on the planet. This protozoa actually contains four important phylum. These are all the four important phylum that we are going to see in this protozoa. The first one is Plasmodiophoromycota, Acrasiomycota, Dictyosteliomycota, and Myxomycota. In this lecture, we are going to see this Plasmodiophoromycota. In detail about Acrasiomycota, Dictyosteliomycota, and Myxomycota can be explained in lecture number 15, 16, and 17 respectively. Now we will see in detail about Plasmodiophoromycota. This and some common features about this four phylum is Plasmodiophoromycota, Acrasiomycota, Dictyosteliomycota, and Myxomycota are commonly called as slime molds, because almost all protozoans are like slime molds only. These four members are this four phylum are commonly called as slime molds. If you go in detail about Dictyosteliomycota and Acrasiomycota, which lacks plasmodia, because the special feature of this protozoans are few of the members it can produces plasmodia. So plasmodia is nothing but multinucleate mass of protoplasm is known as plasmodium. So this Dictyosteliomycota and Acrasiomycota produces no plasmodia. That's why it's called as cellular slimy molds. This Plasmodiophoromycota and Myxomycota, they produce plasmodia. That's why it's called as plasmodial slimy molds, or true slimy molds. So this is also like we can expect next questions or maybe some other kind of questions in exams, like the presence of plasmodial stage is the important feature, or the presence of plasmodial phase is the common feature in which fungi, Plasmodiophoromycota, Myxomycota, or some other fungi they will give. Generally they will ask only Plasmodiophoromycota. The production of plasmodium in the life cycle of clubroot of cabbage or clubroot of crucifers, that is a Plasmodiophoromycota, is one of the special feature of this fungi, that is Plasmodiophora. So which means there is no other fungi can produce plasmodia other than protozoa. No, there is no other fungi belonging to Oomycota or maybe Zygomycota or Ascomycota or Basidiomycota. They won't produce any plasmodium. Only this Plasmodiophoromycota and Myxomycota group of fungi produces plasmodia in their life cycle. But the repeated questions are like presence of plasmodium is a special characteristic feature or the characteristic feature of the which of the following fungi. So they will give four fungi, but definitely Plasmodiophoromycota will be definitely there. That is Plasmodiophoromycota will be definitely there. So this is one important feature. Again, in detail further categorization, this Plasmodiophoromycota generally known as endoparasitic slime molds, because they actually grow inside the root hairs or the root cortex. That's why it's called endo. If it is producing outside, it's called ecto, right? So endo means inside. That's why it's called as endoparasitic slime molds. Acrasiomycota generally called as acracid cellular slimy molds. Dictyosteliomycota are generally called as dictyostelide cellular slimy mold. The Myxomycota is called as a true cellular slimy mold. In the entire this protozoa, Plasmodiophoromycota and Myxomycota are the two important phylum that we are going to see and concentrate. If you guys ask me the most important one, I will suggest Plasmodiophoromycota. In the previous year question papers are Mostly everyone used to concentrate on this Plasmodiophoromycota only. This Myxomycota very less, but we're going to see in detail about everything as I mentioned in the earlier slide. Every single phylum that we are going to see in detail, again this Plasmodiophoromycota, Crassiomycota, Dictyosteliomycota and Myxomycota in detail. So this is the general classification: Plasmodiophoromycota, Acrasiomycota, Plasmodiophorm endocellular slimy molds, acracid cellular slimy molds, Dictyosteliomycota, Myxomycota true cellular slimy mold. And for exam point of view, you guys just remember this: phylum always ends with mycota, subphylum always ends with mycotina, class always ends with mycetes, order always ends with else, family always ends with ACA. So this is some exam point of view that we need to keep it in. Generally sometimes they may ask family always ends with, order always ends with, so some kind of questions we may expect. Now we are going to see in detail about this Plasmodiophoromycota, that is Plasmodiopora. Now we'll see the phylum Plasmodiophoromycota. Let's start with general characters and all the general features. Then we will enter into in detail about the genus level. The general characters of Plasmodiophoromycota includes generally this Plasmodiophoromycota are generally called as slimy molds, which are microscopic obligate parasites which actually grow on freshwater algae and fungi. The thallus is actually naked and it produces a holocarpic plasmodium. Here, the production of plasmodium in the life cycle of clubroot of cabbage is one of the special feature. So in exams also they will ask like in which of the following pathogen can produce plasmodium in their life cycle. That is a Plasmodiophoromycota, that is a Plasmodiophora brassicae. So this is one of the important one. So let me tell you what is plasmodium. So plasmodium is a multinucleate mass of protoplasm is known as plasmodium. So if the protoplasm is in multinucleate, then it is called as plasmodium. So except the reniform stage, the entire life cycle will happens within the host. Now I am going to tell you the life cycle also. There you will understand what is the reniform stage and all the things. The entire life cycle happens in the within the host only, in the root cells and cortical cells, except reinfection stage, not reniform stage, reinfection stage that happens in soil. So before reinfecting, sometimes it will swim in the water, so that happens in the soil. The remaining entire life cycle happens within the host, especially root hairs and as well as cortical cells. So the cruciform type of nuclear division can also be seen in the life cycle of Plasmodiophoromycota. This is also one special feature because the cruciform type of cell division can observed in no other fungi, no other classes of fungi except this Plasmodiophoromycota. That is also the special feature. Cruciform divisions means continuous divisions or repeated kind of divisions can be observed in the life cycle. So zoospores are generally biflagellate with the anterior whiplash type, unequal, one is short and one is long. So such kind of flagella is known as anisocant. In the previous lectures, the types of flagella and the types of zoospores and zoospore morphology also have explained. There are four types of flagella: posterior whiplash, anterior tinsel, biflagellate with anterior whiplash type, one is short, one is long, anterior tinsel and posterior whiplash. So like chytridiomycota, hypocytridiomycota, Plasmodiophoromycota, oomycota, such kind of fungi can produce this kind of flagella, where I have already explained in the types of flagella as well as zoospore morphology also. If you miss the class, you please refer that, then only you will understand what is this kind of flagella and all those things. So the production of this unequal lengths of whiplash flagella is the special feature of this Plasmodiophoromycota. The life cycle is generally happens generally two distinct phases of plasmodium can be observed. As I told you, multinucleate mass of protoplasm is known as plasmodium. So two distinct plasmodial stages can be observed. So generally this plasmodium possesses amoeboid features and can produce pseudopodia. Pseudopodia means like constructions that we have already, that is going to explain in detail about in the later slides, like in Acrasiomycota and Dictyosteliomycota, that engulf and host cytoplasm in the by actually the phagocytosis. So this plasmodium is the two distinct phases that we can observe in this life cycle. And maybe it contains chitin or cellulose in the cell walls. Then the other important thing is the symptoms are produced mainly due to the hypertrophy and hyperplasia symptoms. So hypertrophy and hyperplasia means, hypertrophy means abnormal enlargement of host cells. It is called hypertrophy. Hyperplasia means increase in number of cells or multiplication of host cells. That is the main reason for the production of symptoms. Why this hypertrophy and hyperplasia happens? Due to the production of growth hormones or excessive production of growth hormones. The cells like a bulged or overgrowth or overproduction of cell size or over enlargement of cell size happens. That is the due to the production of the growth hormones. So the infection results hypertrophy and hyperplasia. I told you what is hypertrophy and hyperplasia. And in the walled stages, this Plasmodiophoromycota, that is confined to the production of juice pores, and the juice sporangium, juice pores, and all those things will happen inside the host only. So that all those things that we will see in the life cycle. So the best example is the club root of cabbage or club root of crucifers, which is caused by Plasmodiophora brassicae. So we are discussing a lot about Plasmodium, Plasmodia. Let's see what is Plasmodium and what are the different types of Plasmodia are there. So Plasmodium means multinucleate mass of protoplasm is known as Plasmodium, as I told you. I given a little bit extra information. So this Plasmodium is actually a naked motile. It can locomote. Multinucleate mass of protoplasm that enveloped in the plasma membrane is actually present in the plasma membrane. Instead of naked, it contains a gelatin sheath. Outside it contains a gelatin sheath that actually feeds in an amoeboid fashion. However, the amoeba moves like a twisting or gliding or like a limax. However, the limax will move the same way this amoeba also will move in amoeboid fashion. It feeds the gelatin sheath that feeds actually in the amoeboid fashion. So this Alexopolus and MIMS categorized 4 different types of Plasmodia generally, but we are going to explain only 3 different important types. And the fourth one is almost resembles like this protoplasm and protoplasmodium and aphanoplasmodium only. So protoplasmodium, in proto means first, aphanoplasmodium, aphanis means invisible. In phaneroplasmodium, phaneros means visible. All are Greek words only. Let's see in detail about one by one. With slight variations only will found from one plasmodium to another plasmodium. For example, if you see protoplasmodium, it is the most primitive type. Aphanoplasmodium, initial stage it is primitive only, later it will be slowly start to develop like that. We'll see. The first one is a protoplasmodium. As I told you, it's a most primitive type of plasmodium we can observe in the microscope. And there is no wings and there is no special deviations or differentiation that we can observe in the protoplasm. We cannot distinguish this protoplasm, and the size ranges from less than 1 mm only. It always lies, and it does not show any rhythmical reversible streaming of cytoplasm. There is no reverse streaming of cytoplasm can be observed. So the best example is echinosteliales. Second one is Aphanoplasmodium. It is an inconspicuous, flat, transparent structures with a open network of plasmodial strands. So, in detail explanation that was given in the Myxomycota group of fungi about this plasmodium, types of Plasmodium, pictures along with their detailed explanation as given in Myxomycota. This is just you guys just remember that what are the different types of Plasmodia are there. There are mainly 3 different types: Protoplasmodium, Aphanoplasmodium, Phaneroplasmodium. These are all the 3 different types, right? So generally, Protoplasmodium shows no rhythmical reversible streaming protoplasm, but when it comes to Aphanoplasmodium, it shows rhythmically reversible streaming of protoplasm, which means reverse streaming, forward streaming, backward streaming, that kind of streaming of protoplasm that cannot observed in protoplasm, but that can be observed in this aphanoplasm. So this plasmodia is actually gelidified fluid regions. Veins are not that much conspicuous or differentiated. We can generally observe this in Steminomycetidae. The third one is Phaneroplasmodium, which is actually a large, massive, fan-like structure. So don't worry about that. In Myxomycota, I have given a detailed explanation along with the diagrammatic representation also. There you guys will observe what is this Plasmodium and the different kinds of plasmodial arrangements or organizations. So generally this is yellow in color, which contains granular protoplasm, gelidified fluid. The veins are conspicuously differentiated, and it also shows rhythmically reversible streaming of cytoplasm as like of Aphanoplasmodium. Generally we can observe it in Physareals. The fourth type of Plasmodium shares the common features of Aphanoplasmodium and as well as Phaneroplasmodium that was first discovered by Ramello in 1976 in trichales. So these are all the different types of Plasmodium in Plasmodiophoromycota. The phylum Plasmodiophoromycota contains three important genus. First one is the Plasmodiopora, second one is Pongospora and Polymixa. Now we will see in detail about one by one. The first one is Plasmodiopora. So the genus Plasmodiopora, generally the Plasmodiopora is a group of obligate parasites that known to attack higher plants. The best example is clubroot of cabbage, which is caused by Plasmodiopora brassicae. When we see the DNA analysis, this Plasmodiopora is not that much closely related to Mycota and Straminophila, but it is more closely related to Myxomycota group of fungi. Later, this Myxomycota came into divisions. Now we will see the life cycle of clubroot of cabbage pathogen or clubroot of cabbage. In the life cycle, we are going to see the only important basic points. I'm not going to explain in detail about all the life cycle because we have plenty of life cycle in upcoming fungi also. So I'm going to give just important features or important points in the life cycle. That's it. So when it comes to the history, this was actually first discovered by the Russian scientist. The life cycle of this clubroot of cabbage pathogen was first discovered by Russian scientist M.S. Voronin in 1876-77 and named the pathogen is Plasmodiophora brassicae. It is actually a soil-borne pathogen, obligate endoparasite. Endo means which actually the life cycle and the phases which happen inside the roots or inside the host. That's why it is a endoparasite. In 1870s time, this clubroot was a very serious and very dangerous disease in Russia, and the Russian government gave an open statement that whoever will identify the reason and along with the management practices, they will get a prize. So like that they have announced openly, and many scientists actually tried, but nobody could figure the exact reason behind it. And in 1870s, in the starting of 1870s, Voronin came into action, and 1874, at the time of 1874, he published his first work, and 1877 he completely identified the reason along with the management practices of this pathogen, and he is well renowned for discovery of a clubroot of cabbage life cycle along with potato wart. One more pathogen is there, Synchytrum endobioticum. That life cycle is also discovered by this M.S. Voronin only. He is a student of Anton de Berry actually. And this disease was famous with various kinds of names. Finger and toe disease or Hamburg disease in England, club root or clump foot, club foot in USA, Kopostnikova Kela in Russia, Clutch and Klopdescola coles in Germany. So these are all the different names in each and every country are using. So in exams we expected like finger and toe disease, which disease is actually the synonym of a finger and toe disease, clubroot of cabbage. So this question was I already asked in many times. So just go through about the other names as well. So this disease was very severe in acidic and as well as poorly drained soil. This disease was very severe. Coming to the taxonomy of the pathogen, domain Eukarya, kingdom Protozoa, phylum Plasmodiophoromycota, class Plasmodiophoromycetes, order Plasmodiophorales, genus Plasmodiopora, species Brassicae. This is the taxonomic classification of the pathogen. When we see the symptoms, as I told you, club root, the name itself indicates, we can see, and the first will start with the upper symptoms. So if you see the upper symptom, generally wilting and yellowing, stunting can be observed. Wilting of leaves, yellowing and stunting. Why wilting, yellowing and stunting? Imagine if the roots have no capacity to absorb water, how the nutrients and water will translocate to the roots to shoots. So if there is no translocation and all the blockage of required minerals, water and all the kinds of necessities, so automatically what will happen? Plant will wilt. Yellowing of leaves will happen because of no chlorophyll. Slowly it starts to stunting. This is a general symptom. So then what if you observe the roots, the clear symptoms can be seen like hypertrophy and hyperplasia growth or bulging of roots or some kind of nodule like or lobe like structures can be seen. These are all due to hypertrophy and hyperplasia. The pathogen forced to produce excess amount of growth hormones, so that what will happen? Abnormal enlargement of cell, increase in number of cell divisions will happen due to this kind of symptoms we can observe. When it comes to the life cycle of Plasmodiophoromycota, I'm not going to explain every single step. I'm going to tell you the simple, common, important features about the life cycle. So here the life cycle of Plasmodiophoromycota happens in divided into two phases. Primary phase, that is a haplophase, occurs in a root hair. We can see here primary phase that happens in root hair. The secondary phase, otherwise called as the diploid phase. The name itself indicates in the phase 1 all are in haploid in condition. The second stage it will become a diploid in condition. So it happens in the cortical cells of the root. These are all the cortical cells of the root. So let's start with the phase number 1, that is a primary phase. Again, this primary phase is divided into subdivisions for our easy understanding. The first one is a primary phase, otherwise called as haplophase, that actually occurs in the root hair region. For our easy understanding, again it was divided into germinating of spore. So generally this juice spore is there, right? So this germinating juice spores come in contact with the root region of the plant. So here this root region of the plant, what will happen? This flagella contains this juice spore contains flagella, right? This flagella gets shredded, which means it releases or it takes the flagella out. So it starts to enter into the root cells or the root region of the plant. So this juice spore is generally pear shaped juice spore. It contains of a flagella. So generally the flagella type of Plasmodiophoromycota is anterior whiplash type, right? One is short and one is long. It shreds the flagella and starts to enter into the root region. So once it shreds the flagella and enters into the root region, that zoospore is known as myxoamoeba. So this flagella becomes actually amoeboid. So the terminology used when it enters into the root region is a myxoamoeba. That myxoamoeba enters into the root hair by dissolving the cell wall. After once enters into the cell wall, when it enters into the root hair region, this uninucleate myxoamoeba enlarges. Again, now it's enlarges. We can see here compartmentalization happens. Again, it will slowly divide into this multinucleate mass of protoplasm. Slowly starts to divide into different segments. A small kind of uninucleate spores or uninucleate. We can also call it as juice spores. When it's released, it is called as juice spore. So then uninucleate segments, cruciferous type of divisions means repeated divisions. In the general characters itself we have discussed, cruciferous type of divisions is one of the specific and important feature when it comes to this Plasmodiophoromycota. So after this myxoamoeba, it starts cruciferous division, means continuous division. The same cell divided into many number of cells, that is called a cruciferous type of division. That results in the production of a multinucleate mass of protoplasm. Protoplasm is known as plasmodium. This multinucleate mass of protoplasm is known as plasmodium. In the sexual reproduction, what will happen? This that plasmodium actually contains 4 to 8 compartmentation, or 4 to 8 cells will be formed in a juice sporangium. You can see here, this is generally it's a juice sporangium. 4 to 8 cells. See, this all things will happen in a root region. Imagine this all things will happen in a root region, root cortex region. So this is all 4 to 8 nuclei, that is a daughter cells are formed. When this root was broken down, or when this juice spores were released out, before releasing each nuclei, it forms a cell wall around itself and releases like a juice spore, which means from each infection of 4 to 8 daughter nuclei are produced. The daughter nuclei later converted or functions as a juice spores. This is a phase number 1. When it comes to the secondary phase or diplophase, the name itself indicates diploid phase. The shredded the released juice spores then start to infect the cortex region. After root, it enters into the cortex region. So through root hair, the juice spores on the root hair that enters into the cortex region. Again, here also it shreds the flagella and enters into the cortex region. So in the cortex region, mitotic divisions will happen and a secondary plasmodium is formed. That's why two plasmodial stage were formed. I told you in general characters of fungi, right? Two plasmodial stages was formed. One first plasmodial stage in the root hair infection, second plasmodial stages in the cortex region infection. So that's why two plasmodial stages are formed. In the first phase, it is a haploid. Second phase, it becomes diploid. After mitosis, this multinucleate plasmodium becomes the multinucleate secondary plasmodium, and this again hypertrophy, where this cells, wherever this multinucleate secondary plasmodium produced, again it starts to produce various kinds of growth hormones. That is the reason where hypertrophy and hyperplasia will happen. That what we are seeing outside the symptoms like abnormal enlargement of cells, abnormal enlargement of number of cells that can we are observing the outside. So where this secondary plasmodium in the cortex region starts to infect or once it become the secondary plasmodium, hypertrophy and hyperplasia divisions can take place, and the later stages karyogamy and meiosis also takes place. Here this is in the diploid stage now. This diploid nuclei which forms which goes through a special phase is known as eukaryote phase. This is one of the special phase. They will happen, they will ask like eukaryotic phase can be observed in the primary phase or secondary phase of club root of cabbage life cycle. In the secondary phase only, this diploid nuclei pass through a special phase during the formation of spores, that is called eukaryotic phase. Eukaryote in the sense it become very thick, so it is a special kind of phase. And later what will happen, several haploid resting spores are formed and liberated in the host. Again the life cycle continues like this. The mode of spread is a primary is a resting spore, secondary is a juice spores. The favorable conditions for the disease development is 20 to 24 degrees centigrade when the pH is 6.5. Around 5.2 this is very favorable. When the pH increases, automatically we can manage this disease. So coming to the management aspects, use of disease-free seedlings and the removal of affected roots. Application of lime, it's a very important because this disease is very severe at acid soils and as well as poor drainage facilities or aeration conditions. So if we at 5.2 is acidic, right? If we increase the soil pH to around 7.0 or 7.2 or maybe in between 6.5, above 6.5 to 7.0, we can manage the disease. For increasing the soil pH, lime can be recommended. So the application of 2.5 tons of lime for 2 weeks after transplanting is the best management practice for the management of the disease. This was also given by Voronin. And also soil drenching with copper oxychloride will be helpful, and soil fumigation with methyl bromide 1 kilogram for 10 meter square also can be useful, and seed treatment with captan therum at the rate of 4 gram followed by trichoderma viride seed treatment at the rate of 4 gram per kilogram can be recommended. During nursery stages we can drench with carbendazim or chlorothalonil at the rate of 0.1%. So this is the total life cycle. So juice spore starts to infect the root region and it shuts the flagella, enters into the root, inside the root. So the entered juice spore is known as myxoamoeba. Later compartmentization, slowly compartmentization starts. Multinucleate protoplasm is formed. That multinucleate protoplasm again produces this juice sporangia in the root hair. It produces 4 to 8 uninucleate spores. Later compartmentization again starts and they starts to like a juice spores. Once this juice spore starts to infect this cortical cells, so it forms a specialized phase that is called eukaryotic phase, where the hypertrophy and hyperplasia will happen. So in the formation of this during forming the spores, they it goes through a specialized phase called eukaryotic phase, and again the juice spores are released. The life cycle continues like this. So the next genus is Spongospora. So the Spongospora mainly consists of 3 important species. They are also obligate endoparasite. The first one is a powdery scab of potato, which is caused by Spongospora subterranea. So previously called as Spongospora subterranea, former species subterranea, and crook rot of watergrass, which is caused by Spongospora nastuti, formerly called as Spongospora subterranea, former species nastuti. This genus from Spongospora generally commonly act as a vectors for transmitting the plant viral diseases. So these are all the different kinds of viral diseases that are spread by Spongospora: potato mop top virus, beet soil-borne virus, beet virus Q, and broadbean necrosis virus. These are all the different kinds of viruses that is transmitted by the species genus Spongospora. And coming to the life cycle, I'm not going to explain in detail. Whatever the life cycle we have observed in the clubroot, same kind of life cycle we also observed here. But the symptoms are like on the tubers, powdery pustules can be observed, and clumps also can be observed. If you cut open the tuber with a little bit decaying of tubers can be observed, and the remaining life cycle is almost as like of Plasmodiophora only. In the root hairs the plasmodia is produced, again juice sporangia, all those things, whatever the Plasmodiophora observed, the same thing we can also observe in this potato wart pathogen. And the next one is Polymyxa, genus Polymyxa. The fungi is Polymyxa graminis is a very important one, which actually transmits various plant viral disease, that is a wheat soil borne, barley yellow dwarf mosaic, barley yellow mosaic, peanut clump, rice necrosis, stripe necrosis, wheat spindle streak, beet necrotic yellows, Polymyxa beta generally transmits beet necrotic yellowing virus. So these are all the different kinds of viruses that is transmitted by the genus Polymyxa. In addition to the damaging the crops, some species like Spongospora subterranea, Polymyxa beta, Polymyxa graminis act as a vectors. So in the Protozoa, especially Plasmodiophoromycota, this Polymyxa and Spongospora are the important genus that act as vectors in transmitting the plant viral diseases. So these are all the list of important diseases that is transmitted by the different that is Polymyxa and Spongospora. If you see, these all are transmitted by Polymyxa graminis, barley, mild mosaic virus, barley, yellow dwarf oat. All those things are transmitted by Polymyxa graminis. When it comes to Polymyxa beta, Polymyxa graminis, Spongospora subterranea, all put together, they are important things. But one very important one, generally I used to observe in exam, potato mop top virus. Potato mop top virus is the virus that is actually transmitted by Spongospora subterranea. It's a very, very important one. Potato mop top virus is transmitted by Spongospora subterranea, something like that we can observe. For further reference and more information, student can refer my book, A Vision into Plant Pathology, complete student version. Further doubts and guidance, student can reach us at www.geekyresearch.com. Stay geeky and stay tuned. We are Team Geeky Researchers.